When we, and Jason, I apologize if Jason's already gone over this, this will be kind of a little review. But so the minor allele frequency is the frequency at which the least common allele occurs in the population. And so for common variations, that's defined as having a minor allele frequency of greater than or equal to 5%. So low frequency variants are in between 0.5 and 5%. And if we talk about rare variants, it's really variants with a minor allele frequency of less than 0.5%. So as you've heard a really wonderful overview of what we found with genome-wide association studies for the identification of common variants. But actually, most of human genetic variation is rare. So about 67%, the genes have a minor allele frequency less than 0.1. So this is a really important aspect of human genetic variation that I think is very important to understand. Oh, sorry, if anyone wanted to spend a little more time on this, I'll put it back up. So, and I think Jason showed this slide previously, and so just to remind you about this landscape of genomic variation and the methodologies for studying this. So for disorders that are Mendelian, where there's basically a specific variant that's causally related to a disease, that is not what we have going on in psychiatric genetics, so let's be clear about that. But these have really, this landscape has really been very well studied with linkage studies in families. And so a lot of these, in fact, the majority of these have already been discovered. So, so we're kind of done over here to some extent. I mean, not done in that we haven't cured these disorders, but in terms of understanding the genetics, we've made a lot of progress. Now, for genome-wide association studies, those are in this common area of the allele frequency spectrum, but each of those, as you've heard, has a low effect size on disease risk, and that's most variants identified by genome-wide association studies. Now, high penetrance common variants are very unusual for common disease, so probably one of the few examples we can think of is the ApoE4 allele, which you heard about from Paul. So low frequency variants with intermediate penetrance, this kind of sweet spot here, this is what we can really focus on with these genome-wide and exome sequencing methods, also with C&V calling. So that's really what I'm going to be focusing on. Now, if you, down here, we have genes that both have a low penetrance and are very rare. So basically, if you try to find something like that, you're going to be probably picking, that's probably not a good idea. So I think we can just not talk about that too much today. So in terms of rare variation and how it is relevant to disease, when we think about rare genetic disorders, there are single gene disorders that are high penetrance. These are really the Mendelian disorders I was mentioning. And then more frequently, we have aneuploidies, which are chromosomal variations. So so for example, Down syndrome, where you have 3 copies of chromosome 21. That would be an example of an aneuploidy. There's also pretty common sex chromosome aneuploidies, like Turner syndrome, where you have only 1 copy of an X chromosome and, yeah, just 1 copy of the X. And so these rare, and then this also includes structural variation, and copy number variation is a form of structural variation, which I'll talk more about. And so these rare genetic disorders can result from alterations of single nucleotides in one gene, or an entire chromosome with hundreds of genes involved, or really anywhere in between. And one of the things that's interesting, when we look across these single gene disorders, so we have these databases. If you're interested in taking a look at any of these, the OHBM database, or Online Mendelian Inheritance in Man, and then there's also a database called G2P, or Genotype to Phenotype. And so if you look in these databases, one thing that's really interesting is that across these rare genetic disorders, in OHBM, 74% have some neuropsychiatric CNS symptoms that are involved in that disorder, even though that may not be the primary manifestation of the disorder. So neuropsychiatric symptoms are very common across these rare genetic disorders, even though many other systems are affected. And we see that the largest contribution of these rare disorders, rare highly penetrant disorders, genetic causes, is for neuropsychiatric disorders that have a high heritability, that have an early age of onset that are associated with impaired cognition and or reduced fecundity, so reduced fertility. And so I'll talk a little bit more about that, but across these, the landscape of neuropsychiatric disorders, there are varying contributions of rare mutations. So I think you've already heard what copy number variation is, and so it's a kind of structural variation in the genome that involves unbalanced rearrangements of DNA segments, typically now defined as 50 base pairs or more. Microdeletions, so these are microdeletions or microduplications of DNA segments. And so the number of repeats varies between individuals in the population. And so one question that's really arisen is what is a benign versus a pathogenic CNV? And so there are now ways of classifying these CNVs. We know that there are specific genomic regions that are more likely to harbor these regions of copy number variation, and they can be called genomic hotspots. And there is, in fact, and if you look across the genome, quite a lot of the genome is actually copy number variable. And depending on how you define it with this more inclusive or stringent map, you can see that it varies across chromosomes, with some having quite a bit more copy number variation than others. So there are some special classes of CNVs. There are rearrangement breakpoints that are mediated by stretches of repetitive DNA, and this is by a mechanism called non-allelic homologous recombination. So that basically what that means is you have segments of DNA that are repeated over and over. They're also called segmental duplications, and because those same base pairs are repeated over and over, the genome, when it rearranges, is likely to get confused and make a mistake, and so so that will cause a deletion or duplication to occur in those particular regions more frequently. And so these are often causes of genomic disorders, and many of them do have a neuropsychiatric component. So I've already told you about what these copy number variations look like in terms of duplications or deletions of DNA segments. And one of the things that I think is really interesting is that there is a continuous spectrum of phenotypic effects of CNVs. And so some of them, in fact, are embryonic lethal, where it's not viable if you have that CNV, ranging all the way to no phenotypic effect or even an adaptive trait. And as I mentioned, there are many CNVs do underlie diseases, and many have a neuropsychiatric phenotype. Now, there's a difference also I want to distinguish between de novo versus inherited CNVs. And so de novo is one that arises spontaneously in a child, not inherited from the parents. And de novo CNVs are more likely to have a deleterious effect, and that's because they're not subject to evolutionary pressure. So you can imagine that if something's inherited, you've got to be able to pass it on to your child, and so anything that's really deleterious is going to be selected out of the population. So as I mentioned, there is a spectrum of neuropsychiatric phenotypes associated with CNVs, and the other thing I should mention is that this is based on what we know. And our understanding of the spectrum of phenotypic variation is really incomplete because it's based on what's been assessed, right? So for some of these, we really don't know. And that's actually, so that's really changing now that we are assessing CNVs and other structural genomic variation in these large-scale population studies like UK Biobank. So, and then these studies, for example, in Iceland, where they've genotyped the whole population. So this is really contributing to a much greater understanding. In the United States, where we're bringing people in based on a clinical ascertainment, we're only really seeing the tip of the iceberg, because we're only seeing people that are clinically ascertained because of some developmental delay. So I think that's a really important caveat to what we know about the phenotypic spectrum. So, disease-based studies of genetic mutations. So there are, as I mentioned, CNVs are more common in early onset disorders, and particularly ones that have an effect on cognition. So intellectual disability, they're also tend to be associated with birth defects, autism spectrum disorder, epilepsy. And so if we look at some of these most common CNVs, and I'm going to talk about some of these in more detail, we can see that the core phenotypes really differ for these specific CNVs. So for the 22q11 deletion, which is my personal favorite CNV, and I'll talk more about that one. But you can see that there is the spectrum of intellectual disability as well as a pretty high rate of schizophrenia, whereas for 15q13, it's a SHANK3 mutation that is primarily associated with intellectual disability and epilepsy. Okay. So, as I've mentioned, there's really been this started about in 2008, this really increased understanding of the contribution of rare variants to neuropsychiatric disorders. And while the majority of cases of developmental neuropsychiatric disorders remain of unknown or what we call idiopathic etiology, the fact that we do see these rare genomic copy number variants that may account for a larger proportion of cases than was previously believed, so probably accounting for about 2% of cases of idiopathic schizophrenia and up to 20% of cases of simplex autism spectrum disorder, by simplex meaning one child is affected in the family. And so the fact that we have this phenomenon where there's one disorder that can arise from multiple different mutations, as well as the converse, where we see one mutation that leads to multiple different disorders, and I think Sven mentioned this in his talk as well, just the spectrum of comorbidity that we see. The fact that we see this phenomenon really provides strong evidence for genetic pleiotropy and also challenges these widely held traditional views that we have of diagnostic categories. So we know there are several lines of evidence for a CNV contribution to schizophrenia, and so there's genome-wide enrichment for rare deletions and duplications in schizophrenia cases relative to controls, higher rate of de novo CNVs in cases, and there's also some association evidence implicating a small number of specific loci. Now, all these CNVs implicated in schizophrenia are rare in the population, but they do confer significant risk with odds ratios ranging from 2 to 60. So I think that's really important when you're thinking about what is the right disorder to study, perhaps, that these CNVs, while they're rare, they do have much larger effects than common variation. So, and I think Sven may have showed this, but did you talk about this study? Okay. Okay. Yeah, so this study I just want to highlight because I think it's a really fantastic study of looking at copy number variation across the genome. And for many years, we knew that these deletions and duplications had an effect, but it really had not been studied on a large scale on a genome-wide basis. And so so what they found, first of all, and there's a number of different ways that you can look at CNVs. First, you can look at in terms of the total distance covered by these copy number variants. You can look at the number of genes affected or the total CNV number or total CNV burden. And so basically any which way you cut it, there was a significantly increased CNV burden in schizophrenia cases relative to controls. And what they found was genome-wide significant evidence for 8 loci at 1Q, 2P, which is the orexin-1 gene, 3Q29, 7Q11.2, 15Q13, and both the distal and proximal 16P11, and then 22Q11.2, which Sven mentioned, is so among these CNV losses or deletions, 22Q11.2 was far and away the strongest association with schizophrenia. Now, interestingly, if you look at CNV gains or gains of genomic material or duplications, 16P11.2 duplication showed an association with schizophrenia, but not the deletion. And so I'm going to highlight a couple of CNVs which I think have been studied in more detail and are really quite interesting. So there are, at the 16P11.2 locus, there are reciprocal recurrent CNVs. So there's both these deletion and duplication CNVs. Now there's 29 genes at this locus, and apparently this tends to have the same, it's basically recurrent in the sense that it's the same deletion or duplication all the time. So there's not a lot of variability in what these breakpoints are. Now here's something that I think is really fascinating. So both the deletion and duplication occur in about 1 in 2,000 individuals. But there are mirror image phenotypes, and there's anthropometric variation that varies. So if you have a 16p1 deletion, that tends to be associated with macrocephaly, so increased head size, as well as obesity, whereas you get the opposite phenotype in those with the duplication, microcephaly and underweight. They're both associated with intellectual disability, lower IQ, and similarly increased risk for autism. But we do see some specific risk for schizophrenia, as I mentioned, that's associated with the duplication and not the deletion. And so these are just some of the papers that pointing out this discovery of obesity due to these deletions, and then the mirror image phenotype with the duplication. And then what's interesting is with the Simon's VIP consortium, they looked at brain structural variation as a function of 16p11 deletion versus duplication. And interestingly, what they found was that the duplication was associated with a significant decrease in surface area, and they found the opposite. The deletion was associated with significantly increased brain surface area. So this was specific to cortical surface area. They didn't see any differences in cortical thickness. And also, interestingly, there appears to be a similar kind of mirror image phenotype associated with the 1Q region, where deletions and duplications are associated with micro or macrocephaly, respectively, and both associated with developmental and behavioral abnormalities. So I want to highlight, this was a study done in the Icelandic population by Carrie and Andreas Meyer-Lindenberg, and where they found, and one of the really important points about this study was this was done population-wide. So these were not individuals that were clinically ascertained. This was done across the population. They found people that were carrying the 15q11.2 deletion and duplication, and what they found were these dose-dependent brain and cognitive alterations. And so, this is a little hard to read on this slide, but they did a neurocognitive battery across people with a 15q deletion, and then compared to, so population controls are shown here, so they're basically z-scored at 0, they're average across these neurocognitive measures, whereas their neuropsychiatric C&B controls, as well as the 15q11 deletion carriers, showed cognitive deficits ranging from 0.5 to 1 standard deviation across measures. And for those 15q deletion carriers, it appeared to be specific to measures of arithmetic, and they had higher rates of dyslexia as well as dyscalculia. So there did appear to be some specific learning deficits in those individuals. And then the other really interesting thing that they did was they took, and they did a kind of focused approach of looking at brain structure alterations in these 15q deletion and duplication carriers. And what they found was this mirror image brain phenotype. And they looked specifically at brain structures that were predominantly affected in schizophrenia, because that was what they were interested in. So in the anterior cingulate, the supramarginal gyrus, temporal lobe, they see reductions in brain structure in the deletion and increases in the duplication. So interesting, mirror image brain phenotypes, but the opposite of what was seen in 16p11. But going along with, it was tracking with risk for schizophrenia being associated with this brain reduction phenotype, which is interesting. Okay. So some key questions, and these are key questions that my lab has been really interested in, I'm just going to mention, and maybe at the end we can talk about some key questions that you're interested in. So these reciprocal C&Bs, and then the investigation of gene dosage effects. So I think it's particularly fascinating that these complex behavioral traits, as well as brain phenotypes, are associated with these mirror image phenotypes. And so can we use these genetic first models to really probe circuit level dysfunction in the brain in a translational manner? And one of the nice things about studying a CNV model is that we are able to model the same genetic defect in an animal. And so that's certainly a challenge when you're talking about something, you know, like a common variant that only confers a 0.0001% increase in risk. Or a polygenic model. I mean, this is, and the genetic background is something that's important to think about. So I'll get to that later. another question is whether there's overlap between the genetic and neurobiological mechanisms underlying the development of psychosis, and something that we're particularly interested in the context of a highly penetrant CNB and in the context of idiopathic illness. And then what are the mechanisms underlying divergent outcomes of neuropsychiatric CNBs? So, as I mentioned, I was going to talk a little bit more about 22q11. So this is actually a particular recurrent copy number variant that we've known about for quite some time. Even before the genetics were known, it was known as velocardiofacial or DeGeorge syndrome based on these characteristic physical features. There's also some associated medical comorbidities like cardiac defects and immune deficiency. And as I mentioned, one of the reasons that we're particularly interested in it is because of this really a greatly elevated risk of psychosis that occurs. And there's also quite a bit of what I call diagnostic pluripotency in childhood. They have elevated rates of autism, ADHD, and anxiety disorder, but the rate of the association with schizophrenia appears to be the most specific. And so a couple things, I'll just highlight a couple of our findings that I thought were interesting in relationship to this syndrome. So this is our work from the 22q Enigma working group, where we did a machine learning classification of 22q11 case control status. And if any of you have been doing any kind of machine learning work in the context of idiopathic psychiatric disorders like major depression, you may have found that those results are very variable across cohorts and don't replicate very well. And so one thing that's kind of cool about studying these CNBs is that we actually get very good classification accuracy. So our overall accuracy, when we use 125 cortical features, peaked at 94%. But there were actually a couple of cortical features that were really weighted much more heavily in this classification algorithm. And so if we just took our top two cortical features, this was cortical surface area in the cuneus and the lingual gyrus, we actually get very good separation of 22q cases shown in green and controls shown here in blue. So again, I think this just highlights the point about increased homogeneity when you're looking at one of these rare disorders. So before we even get to issues of specific genes in the region that may be contributing to the phenotype, we just wanted to ask a very basic question about whether deletion size was relevant to the phenotype. And so this is we have a multi-site consortium, which has been really great, as it's allowed us to collect data on over 1,000 individuals with the same confirmed 22q11 microdeletion. And so for this region of the genome, there are these recurrent segmental duplications, and so most people have, about 85% of the patients have a similar large 3 megabase A to D deletion, but there is some variability in it, and so about 8 to 10% of patients have this smaller 1.5 megabase A to B deletion. And so, as you can imagine, it was difficult to really get a massive, large enough sample to actually study the effects of this deletion size. And so this was from our consortium led by Bernice Moro's lab. And so if you look at IQ, there is, in fact, a significantly higher IQ in those both performance, verbal, and full-scale IQ in those with the smaller A to B deletion compared to those with the A to B deletion. And so it's decreased by 0.3 to 0.5 standard deviations in those with the A to D large deletion. So, of course, we were really interested in whether this was related to brain structural differences. And in fact, it is. Whoops. So what we found here was that if you look at differences in cortical surface area, so this is A to D compared to controls, and this actually looks very similar to the deletion overall. But I just want to call your attention to here, where we're comparing those with A to B compared to A to D deletions. And you can see that there's pretty robust differences, and this was specific to cortical surface area again, so we did not see differences in cortical thickness, where the small deletion was associated with significantly larger cortical surface area. Now, The other really intriguing part of this puzzle is, so this study came out from Mike Owen's group a few years ago, showing that duplications of the 22q11 locus, so a gain of genomic material within the same locus, was actually putatively protective against schizophrenia. And so this was done in a large cohort. And so in their discovery sample, they find that there were no cases of no duplications in their schizophrenia group and about 10 in their control group. And then they replicated this. And so this gives us an overall odds ratio of 0.17, so significantly less than 1. So in other words, if you have a 22q11 duplication, you're significantly less likely to develop schizophrenia. Now, it doesn't mean that having a 22q11 duplication is good for you overall, because they do have other things, but less likely to get schizophrenia. And I just want to point out that finding was actually replicated, well, it's a smaller cohort, but in a Han Chinese population. So it does appear to be a real finding, which is pretty interesting. So this 22q11.2 duplication was just described pretty recently in 2009 as a new chromosomal syndrome. And so what's interesting is that you can have the same, basically the identical mutation, the same duplication in a parent that has a completely normal phenotype, but then they have a child that is severely affected. And so this is from the Simon's simplex collection, where they see an overrepresentation of both 22q11 deletions and duplications in that cohort. But if we take for the duplication alone, there's an estimate of frequency of 0.05% of this duplication in non-syndromic simplex autism spectrum disorder. So it is also associated, when there is a phenotype, it's also associated with language impairment and autism spectrum disorder and broad developmental delay. But as I mentioned, there are lots of people out there with this duplication that have no discernible phenotype. Okay, so we were interested in looking at whether this 22q11 deletion duplication showed a similar kind of mirror image pattern to what we'd seen for these other CNVs. And this is work led by Amy Lin, as a neuroscience grad student in my lab. And interestingly, we do see that. So for cortical thickness, we see that those with the 22q duplication had significantly reduced cortical thickness relative to controls, and those with the deletion. So cortical thickness is relatively increased in the deletion cases. We see the opposite pattern for cortical surface area. And so we see very pervasive and widespread disruptions of cortical surface area in both CNV carriers, but the deletion is largely reduced and surface area is increased in those with the duplication. Now here we z-scored these are the top 10 biggest effect sizes for deletion and duplication. And so over here we're looking at cortical thickness, and this is surface area. And so what I think is interesting is that if you look at the brain regions that are most affected, we see medial orbitofrontal cortex, the cingulate, the bank of superior temporal sulcus, superior temporal gyrus, so brain regions, the precuneus, so brain regions that are really critically involved in social cognition and language. Okay, so I'm going to talk a little bit, so I've talked about copy number variation, so I'm going to talk a little bit about sequence variation, which is studied largely so far. It's been studied with whole exome sequencing. And so whole exome sequencing is a technology that assesses the 45 million, approximately, individual nucleotides in protein coding regions of the genome. So these are exons. And you can thus detect single nucleotide variants and indels. And so I want to highlight that this really constitutes about 1% of the genome. So whole genome sequencing is, of course, the much more comprehensive methodology, 3.2 billion individual nucleotides across the genome. And so using whole genome sequencing methods, you can detect single nucleotide variants, indels, large CNVs, and structural variants. And of course, that gets at the 99% of the non-coding genome. Now, as Sven mentioned, this is work in progress. As you can imagine, this is incredibly, well, there's a lot of data. So, So whole genome sequencing, so some things I want to point out, it can improve the detection of common variants that we've seen in GWAS by statistically inferring SNPs that are not directly genotyped. So as Jason probably mentioned, when you're doing looking at common variants, then you're not looking at every single base pair in the genome, and so you're imputing the ones that were not assessed. So this is actually assessing every single base pair across the genome. And then by identifying specific risk variants within a region, so you can do fine mapping. And it may allow the detection of common structural variants, including CMVs, that you might miss by SNP-based approaches. So it's a trade-off, obviously, but you do need to have pretty large samples in order to have the power to deal with the number of comparisons that you're doing. So I just have some terms here, and I'm not going to belabor these exhaustively, but since the slides are going to be posted, I thought that it would be helpful as a handy reference for later. So these are some terms related to rare variation. And so just a couple terms that I want to highlight. So a loss of function variant. These are genetic variants that are predicted to seriously disrupt the function of protein coding genes. And the PLI score is something I'm going to mention in a bit. So this is the probability that a given gene is haploinsufficient and thus likely intolerant of, so it's basically the probability of loss of function, intolerant of loss of function variation. So genes with a high PLI score are extremely intolerant of loss of function variation. And if you have a low PLI score, like 0.1, then that's tolerant of loss of function. So here's a little pop quiz. What is the difference? And this is actually something that's probably gone over before. So it's the difference between a copy number variant and a single nucleotide variant. Okay, well, some of these are silly. It's A, but okay. So that'll be fun for later. You can go back and see how much you remember. Okay, so as Sven talked about, there are variable contributions of rare and common variants across neuropsychiatric disorders. And what it looks like is that for autism spectrum disorder, these rare de novo, likely gene disrupting mutations appear to be most relevant, more so than they are to some of these other later onset disorders. And so when you think about it, it kind of makes sense, given autism is a very severe disorder and involved in typically very early onset, before the age of 2. And so if you look across, this is from a recent perspective paper by Stephen Sanders. So just looking across these disorders, you can see that, so for GWAS studies, so GWAS studies really are not finding much for autism spectrum disorder. There's one genome-wide significant hit. And so it basically looks like the common variant contribution to autism is not as great as it is to some of these other disorders. Now, of course, there are pretty wide differences in sample size, but even given that, you can really see that there is a difference in the genetic architecture of these disorders. And so for exome sequencing, these protein truncating variants, well, actually, yeah, they didn't see any genome-wide significant hits in this study, but now that the samples are getting larger, they're starting to see, and in fact, this study with a family-based exome sequencing study looking at de novo protein truncating variants, there's actually 65 genome-wide hit. And so in a actually somewhat larger sample with rare and de novo protein truncating variants for schizophrenia, there's only one genome-wide significant hit. So it appears that these these kind of pathogenic single gene mutations are really particularly relevant for autism. Okay. So this study, I just want to highlight because I think it's really interesting in terms of how we can potentially use ultra-rare or very rare mutations to predict effects on cognition and potentially brain structure. So that's not been done yet, but this is from Sebastian Jacquemont's group in Montreal. And so what they did was they looked at the effect sizes of CNVs on IQ in community-based samples. So again, these are not clinically ascertained. These were, this is from the Imogen cohort in Europe and another large-scale cohort. And So there is a prior finding, that non-recurrent CNVs in the aggregate are associated with cognitive impairment as well as reduced educational achievement. And so what they looked at here across cohorts, this was over 2,700 individuals, and so they were basically trying to predict what are the aspects of a CNV that would lead to cognitive impairment, and can we predict cognitive impairment and the degree of cognitive impairment based on various aspects of the CNV. So they looked at size of the CNV, the number of genes in it, and the number of exons in it, so the number of protein coding genes within a CNV. And overall, what they find is that it's really, if you look at common deletions or duplications, the effects on cognition are really minimal. So it's really these rare deletions and duplications, and it's particularly deletions. So duplications have a pretty minimal effect on cognitive functioning, which is something that you might expect, given that having a chunk of DNA missing is likely going to disrupt things more than having a little bit more. So that appears to be generally the case. And then the number of genes in the CNV was also important. But you can see that it was really the size of the CNV was associated overall with a 5-point reduction in these rare deletion cases. And then what they did was a linear regression model, and so they wanted to predict IQ based on a number of different aspects of the CNV. And so the PLI score, the predicted loss of function and tolerance of the genes within that CNV, the score rate for intolerance for deletions and duplications, the number of protein-protein interactions, and the differential stability score of regional patterns of brain gene expression. And so they basically made a score for each gene within a particular CNV. And what they found was the best model was really the probability of being loss of function intolerant and how many genes within that CNV were loss of function intolerant. And that was they could actually predict very well what the decrease in IQ would be. And it was interesting that it was particularly performance IQ that was deleteriously affected by the loss of function intolerance of these genes within the deletion region. Now, this does suggest that the CNV effects are polygenic because the more genes you have within the region, it was contributing more. And so it appears to be attributable to some of these individual effects of each gene. So I think that's important because I think when many of us started working with CNVs, we did have the hope, this naive hope, that probably, maybe there's one gene within that CNV region that's really driving the effect. But as usual with anything related to genetics, it's more complicated than that. And so it does appear to be an oligogenic effect. So that brings me to my next point, which is this question of whether there was a common variant contribution to neuropsychiatric disorder risk in high penetrant CNVs. And so Sven talked about polygenic risk scores. And so the SNPs that are detected in a genome-wide association study are characterized by the p-value, so how much they're associated with the disease, and the odds ratio. And so a polygenic risk score is calculated as the sum of the odds ratio for each variant. And so here you have your Manhattan plot for common variation. And then this would be an example of the population distribution of polygenic risk. So you have your, basically the distribution is shifted over in schizophrenia patients. They have a significantly higher polygenic risk than controls. So some possible models that we can think about for disease risk when we're talking about a large effect CNV. And here, in this particular hypothetical example, it's schizophrenia, but it could really be for any phenotype like autism, epilepsy, intellectual disability. And so one possibility is that it's all about having a deletion that causes schizophrenia. So if that were the case, we really would see no polygenic signal in individuals that have a high penetrant CNV. Then the other possibility is that it's really all about polygenic risk. So that's accounting for all of the variants. And so if that were correct, if that model were correct, then we really would all be polygenic risk and nothing really being accounted for by the CNV itself. The third hypothesis is that it's a combination of having this large effect CNV and high polygenic risk that's contributing to the risk for the disorder. And so there's a pretty standard methodology for calculating polygenic risk. And in studies of schizophrenia, it will typically exclude the MHC region because that has a really strong genetic signal. It's a very complicated region. And then it's essentially a logistic regression analysis where you have polygenic risk on phenotype, and then you covary for principal components related to particularly ancestry. That's an important issue, which I think came up before, in sex. And so I'm not going to talk about our 2211 work because it's not published yet, but I want to talk about an example that this is a study from RNAME that's out on bioarchives now, which I was very interested to see. And so what they do find is that polygenic risk is relevant to disease risk in large effect CNVs. And so what they did was they looked at the contribution of common variants to the risk of severe developmental disorders. And so these were kids, heterogeneous cohort, 6,987 children with a severe developmental delay and intellectual disability. And so these are disorders, so these are disorders that with exome sequencing you could detect a monogenic contribution to these disorders. And so they're really believed to be completely monogenic, that those mutations were causal. But as you'll see, there was in fact a polygenic contribution. So the first thing they did was a GWAS of neurodevelopmental disorder risk. And they really did not find any significant, any genes that reached the genome-wide significant threshold, which is not surprising because there was these other large effect genes. But what they did, the next thing they did, which was really interesting, they did what's called a polygenic transmission disequilibrium test in their study. And so essentially comparing polygenic risk in the parents to that in the probands. And then they compared across patients in their study. They did a replication analysis, and then they also looked at genetic correlations between neurodevelopmental disorder and with other traits. And so actually one term I want to highlight here, if you may see in the literature, the Nagelkirk-Yar squared is the proportion of variance between groups that's explained by polygenic risk. And so you can think about the total variance in a disorder is contributed by the CNV, if you have one, as well as the polygenic risk, plus unexplained variance. There's always going to be some stochastic or error, whatever, stuff we don't understand. But what they found was, in fact, that 7.7% of the variance in risk for neurodevelopmental disorders in this cohort was attributable to common genetic variants. And that's actually very similar to the variance accounted for by common disorders like autism and major depression. Now, just to give you some context, at present, polygenic risk appears to explain about 18% of the variance in idiopathic schizophrenia. That's from the latest PGC analysis. Now, there may also be a sample size power issue here because that sample is now up to, I believe, about 60,000 individuals. But so with this this polygenic transmission disequilibrium test, they looked at the transmission disequilibrium of alleles for traits within these trios, and so they find that the polygenic risk is similar in those affected patients and their parents. This common variant signal was positively correlated with genetic predisposition to fewer years of schooling, decreased IQ, as well as schizophrenia risk. And then they also compared their common variant scores for autism, height, birth weight, as well as intracranial volume. And so from other published studies, and those were correlated with those traits within this developmental disorder cohort. And so what I think is really important here is that phenotypic expression in monogenic disorders, or traditionally viewed as monogenic disorders, is affected by the same variants as the general population. So in other words, the risk for developmental disorder is a combination of copy number variation, this large effect, as well as the genetic background. So genetic background matters, and I think that's important. Okay, so central dogma of genetics. And so in the last part, I'm going to segue into the next talk in terms of talking a little bit about gene expression. And so, let's go back to that. So we have DNA here. And so first there is transcription to RNA, and then translation to a protein. So that's a very simple story. And so again, just to highlight what I've been talking about is the level of DNA in the genome. And so I'm now moving on to talk about RNA and the transcriptome. And so here I just want to highlight a particular methodology that we've been using, which I think many of you may find useful for your analyses. So this is a method called weighted gene co-expression network analysis that was developed by my colleague Steve Horvath at UCLA. And what's really nice about this method, so essentially genes that are functionally co-expressed, genes that are co-expressed that show the same patterns of expression are functionally connected with each other, and Steve has showed this in a number of analyses. But so what's really nice about this is that it provides us an unbiased way of defining genes with functional relationships, and it can reveal the underlying organization of the transcriptome based on the degree of gene neighborhood sharing. And so for those of you that do functional connectivity analysis with imaging, some of these figures may look familiar. So it's really the same principle, where we construct a correlation matrix between all the transcripts, and then it's transformed into a weighted network using a soft thresholding approach. And this network structure has been shown in a number of studies to be highly robust and reproducible. And also that the gene's network position is biologically meaningful. So we can identify groups of co-expressed genes or modules that correspond to key elements of biological function. And then within these modules, we can identify the most central or most highly connected or hub genes. And so what's nice about this method is that it addresses this really big, serious multiple comparison problem that we have in gene expression studies. And so going back to some of the work that Sven highlighted in terms of the problems with candidate gene studies, this has also been a major problem for gene expression studies where, you essentially take your favorite gene and then, voila, it's related to all these things. But, obviously it's really important that we take an unbiased approach for these studies. And so this is an overview of the analysis. And so it's an R package. It's very straightforward to use. So this is the link to if you're interested in taking a look at it. And so essentially the way it works is you construct this gene co-expression network analysis. And so these figures, kind of heat maps, will probably look familiar to you for those of you doing imaging analysis. And so essentially you're looking at correlation as a measure across all the genes, all the transcripts, and looking at it as a measure of co-expression. And then you identify these gene modules. Then these modules, you do a dendrogram, and there's a hierarchical clustering of genes that cluster together. And then these modules can be related to external information. So that's nice because they can be related to basically any kind of phenotype information that you have, clinical data, proteomics, and then you can identify biologically interesting modules and then study these module relationships. And because you have a module that has an eigengene, it's essentially like a principal component, this module eigengene, and so it really helps you to reduce this number of comparisons. So just to show this in a very simple way, you have your module eigengene, and then you can relate it to a phenotypic trait, behavior, a brain structure, and so forth. And so this is just an example from some of the work we did. This is looking at patients with 22q deletion compared to controls. And so, but the main point that I want to highlight here is that you will identify a number of differentially expressed genes, and then that cluster into these modules, and then you can identify module trait relationships. We identified different modules that characterize 22q patients with autism spectrum disorder, and that differentiated them from those with psychosis. And then you can also look at both quantitative as well as categorical traits. So, and then once you have your modules, then of course the question is, well, what do those genes in that module mean? And so you can infer the functional relevance of gene sets by conducting an enrichment analysis, and this is done using a test called a hypergeometric overlap test with gene ontology terms. And so you have a gene ontology, which is a database of what these genes do, and then you can ask as the question of whether your clusters include more genes than expected from groups of genes with established pathways and biological functions. And so there's a gene ontology for biological processes, for molecular functions, and cellular components. And so you can basically look at enrichment of your modules for these terms and get a sense of what these genes are doing. And so just to give you a couple of examples how you can use this type of analysis. So this was, this is work in progress by a postdoc in my lab, Jen Forsyth. And so what Jen is doing is looking at developmental gene expression patterns. And so she's using a database called BrainSpan, which is a postmortem brain expression database. And so there's both microarray and RNA sequencing data across different cortical regions and different stages of development. So they have multiple stages of early fetal development going all the way through adulthood. So it's a great resource if you're interested in doing this kind of analysis. And so essentially what Jen is looking at, the first question she's looking at is how are genes, both common and rare variation in the genome, how does it relate to a developmental expression in the brain? And then the second question is how does the 22q11 genes relate to this common and rare genetic variation, and how does it relate to gene expression in the brain? And so what she found, so first of all, these are autism spectrum disorder studies, these are schizophrenia studies, and you can see that there are some genes that are gene modules that are particularly specific to autism spectrum disorder, others that are common to schizophrenia and autism. And you can see that there are a number of 22q11 genes that cluster within these modules. And what's interesting here is that when you look at what these gene modules are, so this M1 module here is an ASD-associated module, and so it shows a very different pattern, developmental expression pattern over time. So this is going from postnatal early prenatal development, 6 weeks preconception, through age 30. And so what you can see is that these genes are highly expressed very early in development, and then they go down. This module is associated with both schizophrenia and ASD. Oh, and then if you look at what the gene functions are with the gene ontology, this module involves functions like chromatin modification, regulation of gene expression, so very, very basic early developmental processes. This other module involved with both schizophrenia and autism, again, gene expression, RNA splicing, whereas we see a very different developmental pattern for this module that was specifically associated with schizophrenia, where the expression is low early in development, but it starts increasing postnatally going up through adulthood. And this is a module that's involved in synaptic transmission and behavior and learning and memory. So, time? Okay. So, last slide. And this, I think Sven mentioned human-induced pluripotent stem cells, one possible future application of how these things relate to each other. And so this is work I'm doing with collaborator Sergio Pashka and Joaquim Hallmeyer at Stanford. So Sergio is doing this really amazing work differentiating, so basically taking skin cells, we take fibroblasts or skin cells from patients with 22q11 deletions and are then able to, well, Sergio is able to differentiate that, reprogram them and differentiate them into neurons. And he has developed a protocol for, he started out doing these monolayer protocols and has now developed a 3D differentiation protocol, so enabling him to develop these mini brains or organoids. And so just to highlight what we are finding so far, you can see first of all that these neural progenitors and neurons from 22q deletion patients and controls really cluster very differently. And then if you look at the gene ontology for genes that are differentially expressed in 22q11 patients, they are enriched for things like synaptic transmission as well as glutamate receptor activity. And this module was also enriched for genes associated with schizophrenia in other studies, as well as postmortem ASD tissue. So, work in progress, but I think very promising and gives us an avenue in which our brain structural data could relate to neuronal, actually, human-induced pluripotent stem cells that were turned into neurons. So I have some questions here, and I think we focus on the big questions. Really, what are some of the questions that you might want to answer with the study of rare genetic variation in the brain? And with that, I will let me thank all the really fantastic people in my lab who did the work, my dog, and acknowledgments of funding sources. All right, thank you.